Hello everyone, I'm Pranjal, and today I'll be talking about my surf project, dye-filling in the head neurons of Steinernema hermaphroditum, which is done under the mentorship of Dr. Paul Sternberg. So I'll be talking about this project in following headings, not necessarily in the same order. C. elegans is a well-established model that has already been employed to study unique biological phenotypes and behaviors. However, it lacks stable mutualistic relationship with the bacteria. Recently, Steinernema hermaphroditum, a newly discovered entomopathogenic nematode, has been demonstrated to be a suitable genetically tractable model to study symbiosis and entomopathogenicity. As seen in this figure on the right side, you can see that it's both entomopathogenic and has a mutualistic relationship with the bacteria. The bacteria in this case is Caenorhabditis griffini. To further study the neuroscientific perspective of behaviors such as symbiosis and pathogenicity in nematodes, we attempted to stain neurons in this species. We attempted to characterize the neuroanatomy in the head region of the said nematode species and also to assess differences in different life stages. Staining neurons is a proven and easy technique to confirm the identity of neurons and presence of cilia and assay the health of those cilia. Let's talk more about cilia. The nervous system of C. elegans contain 302 neurons, out of which 60 neurons have cilia. That is non-motile. Cilia were previously thought to be vestigial in C. elegans, but now they are increasingly being associated with the sensory system. The cilia can be exposed to the environment via opening in the cuticle or terminate or embed in subcuticle, sheath cell, or the other structures. The neurons can uptake fluorescent dyes like dye-I, dye-O, and FTIC via their cilia. However, the absence of dye filling does not conclusively indicate the absence of cilia, as the precise mechanism of dye filling is not known yet. Ciliated neurons and dye-filling phenotype, speaking more specifically, have previously shown to be fairly conserved across different plates of nematodes. This is how the dye-eye stained images of head neurons in C. elegans looks like. Here you can see there are inner label neurons, the nerve ring, and the amphids, the dorsal triplets, ASK, ADL, ASI, and then AWB, ASH, and ASJ. To stain these neurons in C. elegans, we used a modified version of dye filling that is known as Tong and Berglin's method, which was published in 2010. When we tried to stain neurons in Steinernema using this method, we were not able to get very good results. Hence, we conducted a series of experiments with different dye concentrations, incubation solutions, and duration of staining. As seen here, that 20 micrograms per ml is better than both 10 and 5. Tritonex is statistically much better than M9 and H2O, and 5 to 10 hours is best. We chose 5 hours for the sake of convenience. This is how the final dye filling assay we conducted to stain neurons in Steinernema looks like. We started with petri dishes with well-fed worms on it and suspended the worms in 0.5% Tritonex 100 in M9, washed it 3 times, added dye to have a final concentration of 20 micrograms per ml. We put the incubator with the shaker at 25 degrees, washed it again after post-staining, anesthetized with sodium azide, mounted on agarose pad, and viewed the worms using the microscope. Now, how the imaging looks like. The worms were viewed using Zeiss microscope imager with dye and TIC channels. There can be an exclusion criteria because there can be some dead worms, those were not counted, and worms that were filled with dye, which might be due to evisceration, those were not also counted. Positive results, worms with at least one stained neuron and associated dendrite. They can be a negative result, worms that were deemed alive on observation of neuron and body structure and did not show dye filling. We anticipated few false negatives. Worms that were thought to be alive might be dead with the neurodegenerative process under. Here, I would like to clarify that I'm attempting to define positional homologs and not the true homology that requires genetic screens. I used this method To identify the positional homologs, I used a few anatomical landmarks. There can be an excretory pore, which lies on the ventral side of the worm. Then there can be anatomical structural homologs of C. elegans, development of pharynx, terminal bulb, isthmus, and metacarpus. This is the final result of how stained neurons looks like in Steinernema hermaphroditum. The left side is di filled images and the right side is the DIC. As you can see, we can see IL2 and IL1 neurons. The dorsal triplets, those are ASK, ADL in ASI, and the ASJ neurons. Besides dye filling in head neurons, we also observed dye uptake by excretory pore. This is the gut and the excretory pore. Dye uptake by the excretory pore and the excretory duct in Steinernema hermaphroditum can be employed in future to assay the health of pore cell, duct, and excretory system in future studies. We did not observe dye filling in the infective juveniles of Steinernema hermaphroditum. As you can see, very intuitively, there is no gut filling. Some unknown background staining can be seen. Interestingly, worms that were washed and placed in M9 and H2O compared to 0.5% Triton X100 in M9 significantly exhibited inhibited staining. As Triton is often used to lyse bacterial cell membrane, it might interfere with the gut bacteria staining in worms. Similar to dye filling in the amphids of Plasma trapatum, we did not observe dye filling in ASH and AWB. Steinernema hermaphroditum amphineurons were intermittently stained as compared to inner label neurons. This is an interesting finding because this is very similar to what we observe in different species of heterorhabditis as well. Both of them are entomopathogenic and both of them have mutualistic relationship with bacteria. So both of them are quite similar. However, they are in different grades of nematodes. We anticipate the need to revisit our homology assignment along with genetic screens and neuron-specific GFP expression to validate our results. In coming times, Steinernema hermaphroditum will play a significant role in understanding parasitism and symbiosis. Dye-filling phenotype is conserved in Steinernema hermaphroditum and is an efficient way to identify neurons. Further research is definitely required to deduce the exact mechanism of dye filling in select neurons of nematodes because in some life stages, for example, IGs, in Steinernema and heterorhabditis, both of which are entomopathogenic, there is no dye filling. However, in C. elegans, there is dye filling in amphineurons. So there is some mystery which needs to be solved.
At the end, I would like to acknowledge Paul for inviting me to his lab, Paul and James and Raymond for being amazing mentors, Mark and Sharon for providing me comments, Monkey for introducing me to this amazing project, Stephanie and Wilbur for teaching me lab skills, and the entire Sternberg lab for answering all my stupid questions. And at the last, student faculty programs for giving me this wonderful opportunity. Thank you so much.